Welcome to today's lesson on DNA and the genome. The key questions for today are: what is DNA? What is the structure of DNA? What is the genome? How was the genome studied? Why is it important? The key terms you will need to know are DNA, double helix, bases, genome, sequence. DNA, or deoxyribonucleic acid, is the chemical that makes up the genetic material of an organism. It is found in the nucleus of animal cells and has been studied extensively. DNA is a polymer, which is a substance made up of repeating units. These repeating units are called nucleotides and consist of deoxyribose sugar, phosphate and base. The DNA molecule has a twisted double helix with two sugar phosphate strands held together by base pairs. The DNA bases have the names adenine, thymine, guanine and cytosine, or ATGC. Bases A and T are described as complementary, which means they fit together forming a base pair. G and C also fit together as complementary bases. The DNA bases will only fit with their complementary base, so A always bonds to T and C always bonds to G. Task: describe the structure of DNA. DNA strands are coiled into structures called chromosomes. Sections of chromosomes are called genes, and these contain sequences of DNA that code for a certain characteristic, such as hair colour or eye colour. The DNA base sequence codes for amino acids that make up a particular protein. It takes 3 bases to code for one amino acid. These sections of 3 bases are called codons. Task: how many amino acids does this section of DNA code for? These bases code for 3 amino acids. Extension: what is a codon? A codon is a sequence of 3 bases that codes for an amino acid. The whole sequence of an organism's DNA is called its genome. The human genome has been studied extensively as the Human Genome Project. This project identified and mapped all of the genes present in humans. It included genes that are directly responsible for characteristics and those genes that code for the activation of others. Some genes code for physical characteristics of humans, such as hair colour, hair type, eye colour, or even the presence of diseases and disorders, whereas other genes simply switch on other genes. This makes identifying genetic causes for diseases difficult, as healthy genes may be present, but the gene that activates them is not. This makes it difficult to link certain genes to specific diseases. However, through this project, scientists developed a much better understanding of the genes present in humans. Knowledge of the human genome has many applications in medicine and ancestry. Genes can be linked to diseases and screened for to help people manage conditions more effectively. They can be used in the diagnosis and sometimes treatment of inherited disorders. Sometimes a person's genes determine how successful a medication can be, or if there is a predisposition to certain diseases, meaning preventative measures can be put into place. Studying the genes of humans can also track patterns in migration and movements of people, and can help people discover their ancestors or living relatives.